Welcome back, everyone. I'll start over. The Introduction to Bioinformatics course is designed to give you a high-level overview of different techniques that will allow you to study biological phenomena by analyzing next-generation sequencing data. So when we speak about omics data, we talked about processing. Today we'll talk about the analysis, and then we'll also see how interpretation of these patterns could be available for biological insights in different types of projects. So today we will talk about introduction to statistical analysis and machine learning. We'll take the gene expression table that we already prepared last time, kind of review what we did, and then try to analyze that data to understand what's in it and what does it mean. So first of all, let's talk a little bit about the progress overall. Did anyone have any questions about what we covered last time or where we are in the program? I know that a few of you have been working through the Introduction to Bioinformatics, Introduction to Genomics, Genomics 1, but also Transcriptomics 1. So if you have any questions about those, please post them in the chat. I'll be happy to address them. Let's take a look at the progress on our organization page. So looking at the schedule, we started off talking about bioinformatics and big data. We then continued to talk about DNA and genomics. Last time, we talked about transcriptomics, or an introduction to analysis of transcriptomic or RNA-seq data. And then today, we're meeting to talk about statistical analysis and machine learning. And as you can see down here, some of you I see are working through some of those pipelines, analyzing the pipelines from genomics and looking at transcriptomics 1. So for those of you that have not started yet, we have a weekend coming up, so hopefully you'll be able to go through some of that material there. And remember that here we also have a syllabus tab, and in the syllabus tab you'll be able to find what associated topics as well as courses or assignments we have. So we had Introduction to Bioinformatics here, these two courses included here. We did Transcriptomics 1 review last time, and today we'll talk about how to analyze data using statistical analysis and machine learning. Will be a brief introduction for all of you to kind of familiarize yourself with, but a whole lot more is included in some of these resources attached here. In our next session, we'll talk about how many of the methods that we will briefly touch on today could be executed in R or Python, and where you can practice and develop those skills. It's not going to be a comprehensive kind of immersive experience because we just don't have enough time, but that really will give you a perspective on some of those tools that you can continue using. Now, after that, we will talk about how to apply these skills that you've learned about in this brief introduction to different types of projects. We'll introduce different project examples that you can start working with. And if you have a project of your own, you'll be able to think about it in terms of where to find data, how to plan your analysis, and then how to visualize the results of your analysis and present them to your peers or elsewhere. Let's review what we covered last time before we go on in today's session. Last time we talked about gene expression or RNA-seq, and we spoke about measuring the functional protein coding elements, which are genes or transcripts, and how to quantify the level of expression. So RNA-seq kind of starts from that place where you start generating a structured table, and then that structured table could be analyzed. The table represents a number of observations, many observations, which would be genes or isoforms, and those could be tens or even hundreds of thousands. So when we talk about genes, we're talking about 20 to 25,000 genes. When we talk about transcripts, there's a number of transcripts for each gene. So we're talking about many more. In the Transcriptomics 1 course, you were also introduced to the patient-derived xenograft model, which is a project that studies tumor growth in an animal model. The useful part about that project is that you can actually study the interaction between the tumor and the microenvironment by separating the gene expression into two types of genes, genes that come from the human and genes that come from the mouse. And so by having that data annotated with different gene IDs, you can start thinking about the interaction between the two. Now, in the same way, we can think about differences between conditions. When we speak about differences between conditions, if there are just two conditions, we can look for observations, which again are genes, that have differences in expression that are consistent within each group, and therefore we can compare them using a statistical measurement of how probable it would be to find such a difference by chance, giving us a measurement of statistical significance, or a p-value. So today we'll talk about some of these things, but in the coursework that we have available for transcriptomics, we actually have this information divided up into 3 different courses. And so we started off with Introduction to Bioinformatics and Introduction to Genomics, and then as we went through the course last time, we hopefully clarified how to generate, process, and understand what next generation sequencing data actually contains, what information it carries. Today, we will talk about identifying and studying significant trends and kind of speak a little bit about what to do when we have this big data challenge with a lot of different types of conditions put together into a single data set. How can we deal with that complexity and still extract some meaningful patterns from this data? And finally, you have an optional course on Transcriptomics 4, which introduces you to single-cell transcriptomics. Now, in the sessions, hopefully we can address any questions that you might have and do some so that we're not just speaking hypotheticals and theoretical things, but we can actually do this hands-on. So in the coursework, this is the coursework that we have. And as we mentioned, we already created a table of expression. This table of expression has a sample as a column name and a gene as a row name. And in between, we have a level of expression, which could have some different types of signals. For example, here we see 2 genes. So ENSG stands for a human gene, and ENS-MUS stands for a mouse gene, an ensemble ID. And last time we took a couple of these examples to really see what they mean. Now, when you look at this gene expression pattern, you might start thinking about how does this gene expression represent a condition. And so we'll talk today about how to compare those conditions, but if you just visually see this kind of an expression pattern, you might be thinking, okay, well, here I have ER positive, here I have triple negative. How do these 2 conditions compare based on many observations that I have in these groups? What did we learn last time? We learned the role of gene transcription, talked about different methods of quantification for mRNA or messenger RNA, talked about next generation sequencing as one of those methods, and then started talking about analysis of next generation sequencing RNA-seq data. That includes pre-processing, mapping on the reference genome or transcriptome, quantification of the levels of gene expression, mining of gene expression for sample variability, or sorry, the meaning, so how do we interpret that meaning? And we didn't really have a chance to talk in detail about biological interpretation, but we'll talk about that a little bit more today. So to see that all of you are on the same page, that all of you have understood this material, let's review a couple of questions from this quiz from Transcriptomics 1. So let me ask you a question. Please put your answer in the chat. Which of the following statements regarding transcription is correct? 1, the central dogma of molecular biology describes the two-step process, transcription and translation, by which the information in genes flows into proteins, DNA to RNA to protein. Transcription is the synthesis of an RNA copy of a segment of DNA. RNA is synthesized by the enzyme DNA polymerase. 2, the central dogma of molecular biology describes a three-step process, replication, transcription, and translation, by which the information in genes flows into proteins, DNA, RNA, protein. Transcription is the synthesis of an RNA copy of a segment of DNA. RNA is synthesized by the enzyme RNA polymerase. 3, the sum total of all messenger RNA molecules expressed from the genes of an organism are produced during transcription, with exactly none of these RNA molecules being produced during translation. And 4, more than one answer choice is correct. So what do you think is the correct answer? Okay, Genova is saying 2. Stephanie is saying 4. Anyone else? Irmak is saying 2. Okay, so it looks like the correct answer is actually 4. More than one answer choice is correct. Why? Well, first of all, we know that you are correct about number 2, that the difference between A and B here, or 1 and 2, is that we have the enzyme RNA polymerase, which is more logical because we're talking about RNA. But also, we know that number 3 is correct, right? And number 3 is correct because we know that here it says the sum total of all messenger RNA molecules expressed from the genes of an organism are produced during transcription, which is correct, with exactly none of these RNA molecules being produced during translation. Translation is when proteins are produced from mRNA, so mRNA does not get produced during translation. So number 2 and number 3 are both correct. Let's take a look at another question. Which of the following statements in regards to the central dogma of molecular biology is true? 1, an exon is a long stretch of non-coding DNA found between introns or coding regions in a gene. Gene that contains exons are known as discontinuous or split genes, and as the coding regions are not continuous. Exons are found only in eukaryotic organisms. 2, alternative splicing is a process by which exons or portions of exons or non-coding regions within a pre-mRNA transcript are differentially joined or skipped, resulting in multiple protein isoforms being encoded by a single gene. 3, gene isoforms are various RNAs that can be produced from a single gene. Isoforms are also known as splice variants, referring to the process of splicing or cutting out introns and sometimes skipping exons, thus getting different mRNAs from the same gene, sequence using different combinations of introns and exons. And 4, more than one answer choice is correct. Okay, let me make it easier for you. Oh, okay, Genova is saying 3. So I will make it easier, and I will tell you that the correct answer here is 4. More than one answer choice is correct, but one of these answers is incorrect. So which answer is incorrect? So 4 is the correct answer. More than one choice here is correct. Which one is incorrect? Okay, so Irmak and Genova are saying that 1 is incorrect. Okay, so and that is the right answer. So number 1 is incorrect. Why? because actually exons are the coding and introns are the non-coding. So if we switch those around, that would be correct. So you got it right. Okay, so let's talk about what do we do with the information that this gene expression table contains, and how do we infer biological meaning from this? So to speak about this, we'll talk about statistical analysis and machine learning. Can someone give me an example of a statistical analysis? All right, excellent. So JDev is saying t-test. The question is, can you give me an example of a statistical analysis? ANOVA, right? So analysis of variance. T-test, analysis of variance. Great examples, JDev. And probably you have heard of F-test, T-test, right? There's all kinds of Kolmogorov-Smirnov test or KS test, right? So there's a lot of tests. When we say these tests, we are making the assumption that samples or observations that we have represent some kind of a measurement, and we are testing a hypothesis, right? So really, when we speak about statistics, statistical analysis, we are doing an analysis of statistical observations to understand their significance. And does anyone have any idea of what a statistic is? What is a statistic? An example of a statistic. Measure of central tendency. Okay, any other examples? So measure of central tendency, for example, median or mean, right? So a mean is a statistic. We're not really saying all of the data that we have is this. We're just saying one measurement out of all of our samples, all of our observations is a mean. So a mean is a statistic. What other statistics are you familiar with? Median, yeah. Okay, so mean, median, we also have distribution or standard deviation from the mean, right? So that gives us a distribution of our data. So we can look at different types of single measurements from a number of observations. That is a statistic. So when we talk about statistical analysis, instead of looking at all of our data, we're kind of summarizing the data so that we can understand some general trends. Once those trends are designed, so we have a sense of what those trends are, we can start comparing them. And that is the statistical analysis. So let's talk about this in a little bit more detail. When we talk about these kinds of methods, we are going to be reviewing some of the material that is found in Transcriptomics 2. This course has information about differential gene expression that is based on hypothesis testing. And so hypothesis testing means that we have a hypothesis, we have an idea of what we are expecting to find. And then the objective of hypothesis testing is to test whether this hypothesis is true or untrue, could be proven or disproven by our analysis. And so another form of that analysis of variance, could also be looked at from a standpoint of regression, how can we measure relationship between observations and the variables involved in the experiment design, or how the data was prepared. And also, when we do such analysis, we need to have some considerations that are specific to the type of data that we're dealing with, or transcriptomic data, which means that we have to look at, for example, multiple testing, which affects our statistical analysis, but also we have to look at the relationship between what's in the data, the statistical test, and its biological significance. So we'll try to kind of tie in some of these things together in the first portion of today's session. What you see here on the screen is called a box plot, or a box and whisker plot. A box and whisker plot summarizes our statistics about a data set by sample in a visual format. So here what you can see is the mean, so that is the line in the middle. So if you imagine these to be your different types of samples or conditions, and then you have some meaningful statistics about each one that you can start comparing. So for example, the maximum value, the minimum value, the upper quartile, lower quartile, and the median. You can sometimes plot both the median and the mean on a box and whisker plot. But visually, it allows us to differentiate between the bulk of our data, which is in the interquartile range. So this range represents the majority of the data, and allows us to see how distant from the majority of the data are the lower and the top value. Also, sometimes looking at outliers. So outliers are those that are observations from a sample that are very distant from the rest of the data. So the bottom, the minimum and the maximum value represents 95% of all the data. And if you have some observation that is, much different from the rest, that's going to be called an outlier. So there are different thresholds for outliers. For example, you can say an outlier is a gene that is expressed at many levels higher than the rest of the genes. Okay, so it's some abnormal, unusual observation that you have in the data, which we call an outlier. Does that make sense, Jayden? For example, if you're measuring height of people and you see, in meters, let's say we say 1.8 meters, 1.7 meters, 1.5 meters, and then all of a sudden 3 meters, right? So 3 meters is so different from what you would expect to find, you would call that an outlier. Now, when we talk about gene expression data and we take the raw values, those raw values will have, if you make a box plot of those values, they will look weird. They will look strange, because the bulk of your data is going to be here at the bottom and practically undistinguishable in terms of what's inside. So the bulk of your data is difficult to study. Visually, we can see it in a box plot. Statistically, what it means is if a lot of your values are lower than 1,000, and you have a lot of values that go all the way to 16,000, so 16,000 is 16 times higher than what is under 1,000. And that's why it is difficult to compare such values. So if I have a value that is 16,000 and I have a value that is 500, anything that is 500, 400, 300, the differences between those genes are so little compared to these outliers, it is difficult to make those comparisons. So the majority of your values are down here, and the outliers are skewing the distribution of your data. So the skewness of your distribution makes it difficult to run statistical analysis, because there's such a disbalance in the scale of some of the gene expression and the bulk of your data. Does that make sense? Does anyone have any questions about this? Okay, so to address this problem, which is a common problem with biological data has this non-normal distribution. One of the ways to address this challenge is to change the scale. So this is our scale. The Y is the scale for the raw data. And we can change this scale, for example, to logarithmic scale. Logarithmic scale makes distances between very high and low values smaller. And so when we convert our data, as you can see here, we have the raw data, here we have the logarithmic scale data. When we convert it to the scale, all of a sudden the distribution is more normal. And so that's why this is called log scale transformation. And this kind of distribution is called log normal distribution. So the log normal distribution means that if you convert it to logarithmic scale, it will look more normal. So if we study this distribution, here we have just a summary of some of these statistical properties, but we can also specifically look at distribution. How do we do that? We can look at a histogram. So here's a histogram of the same sample. This is the normal, and this is the log scale transformed. So what you see here is a histogram. Who knows what each bar here represents in a histogram? What does a bar represent in a histogram? Okay, exactly. Counts of how many genes, in this case, we have in this bin. So this bin says between 0 and 70. This bin is 210 to 280. So we have these different bins, and these bins contain the number of observations. In our case, observations are genes, and so the majority fall in this first bin. But this right here represents how many observations we have that are farther and farther away from the bulk of our data. So the bulk of our data means how many, or the count of observations within this range. Okay, I'm sorry, Jigar, that you're having some issues. Maybe it's best to use a computer to participate in the session so that you can also be involved in the discussion. But as you can see, if we transform this data to logarithmic scale, what changes is the scale, giving us a better view of the bulk of our data. So now the bulk of the data is right here in the middle. So that's why it is useful to transform our data to logarithmic scale. Another important aspect of data transformation before we start statistical analysis is to understand the distribution and align the means of our data. So oftentimes this is referred to as scaling or normalization. And an important part for gene expression data, because many of our downstream analyses are going to have assumptions about the data. And the assumptions are typically that the distribution is normal and that the means, especially between our different samples, different groups of samples, are going to be aligned, right? So for example, if I have a lot of observations that are from 0 to 10, and then a lot of observations are from 10 to 20, right? So if I have this kind of a jump, the skew between those observations, so 7, 5, 8, 9, etc., and then the other ones are 3, 5, etc., right? So I have this skew, this jump. If I want to compare those two, anything I measure is going to show as different, But in reality, because of the nature of biological observations, the way that they are measured and who measures them can play a major role. Even though biology itself might not be so different, I would like to make sure that the data is not influenced by these technical artifacts. So that's why we can use quantile normalization, and that allows us to better understand the biological meaning of our data, right? Again, think about it as not a universal measurement like a weight or a size. More think about it as a representation of a data that has dependencies on many different aspects, which are typically noisy and hard to control. So when we look at this data, we understand that the low gene expression that we have here at the bottom of this histogram really does not carry a lot of biological information because it makes it difficult to distinguish between the low expressed genes. We also have the bulk of our data, and the bulk of our data typically represents genes that are responsible for the normal function of a cell or a biological organism. And then we have the significant signal. The significant signal that we have about differences is where we will find the most significant and statistically valuable information. And so when we do our quantile normalization, this kind of an exploratory analysis will help us establish a threshold. For example, in this case, the threshold is around 5 because we can see the bins here are around 5. And so we can really keep this information non-normalized and this information normalized so that we account for assuming, our assumption is that both of these living organisms will have normal gene expression overall, and only some genes will have significant differences. So if we compare two examples here, this is an example where we have non-normalized data. So the differences in non-normalized data might lead us to think that this group of samples is very different from this group of samples, even though the noise within each one kind of is eliminated if we do quantile normalization, and we can see that there's not a lot of statistical differences between the two groups. In addition to that, this kind of transformation is important because we have groups of samples. So if we have groups of samples that each have a gene expression observation about each one, we are assuming that biological replicates are going to represent ideally the condition that we are studying. So if we have two samples that are from the same condition, for example, both of them are ER positive, they could still be very different because we captured the data at different differentiation state, or we captured the data in different biological condition or even time. We still expect the bulk of our data to be, sort of comparable between the two samples. And so what you can see here are how those are comparable when we look at correlation between the genes in those two samples. So to really practice this a little bit, let's try and do that in Excel. How many of you have Excel available? Okay, so I think it's really easy to view this in Excel. I'm just going to give you a link to a data set that hopefully all of you will be able to take. All right, let me know once you have this data set downloaded and opened up on your computer, if you do have Excel available, and we'll go through just a very simple illustration so you will see how these plots are generated and how it really represents the different data points and the different conditions that we have in our data. So let me know once you have this file downloaded and open on your computer in Excel. All right, thank you, Mark. Anyone else? I'll just give it a couple more minutes for everybody else to download and open. All right, great, thank you, Prashant. Okay, well, here's the data set, and let's take a look at what's inside. We have here two groups, non-malignant and cloud and low. So these are two subtypes of breast cancer. Non-malignant means that it is a tumor, but nevertheless, it's not a very malignant tumor. And here we have cloud and low, which is one of the subtypes. So inside each one of them, we have the gene expression in raw data. So to create a box plot, we can just select all of these and say insert, and here you have this box and whisker plot option. This is an Excel on a Mac, so it might be a little bit different if you're using a Windows computer. But you can see here how the data is different between those different samples. And as we saw in the example in the presentation, this is really not very informative. So here we can just change this a little bit. Okay, so we can see here our different samples, and the samples are not very easy to work with in this format. So the first thing that we can do is we can convert this data, I'll just move this right here, to logarithmic scale. Now, when we have logarithmic scale, we cannot take a logarithm. Okay, so before we do this, actually, this data set contains some NAs. And so we want to first eliminate those NAs from our data, right? So here I can just search for NAs. There are different ways that you can change NAs. One way is you can, for example, change them just by deleting the full row. You can use regression to fill in NAs. But for simplicity's sake, in our case, right, we're not really going to do a very technical job. What we'll do is we'll just do find and replace. So we'll just find all of our NAs and we will replace them with a 0. Okay, so now we don't have any NAs. And just technically, this will allow us to continue with our exercise. Let's put this back. If we want to change this data to logarithmic scale, we have to run a simple function. So what we'll do is we'll just copy this over. And here we'll just say that we want to take this cell, but we'll change it to logarithmic scale of this plus 1. So why are we doing plus 1? Because we want to make sure that we don't try and take logarithmic scale of 0, which is, we won't be able to do that. So now we can just stretch this across. Oh, sorry. So we can stretch this across. all of our samples. Okay, and don't need this. And now we have our logarithmic scale. So let's try to see what is in the logarithmic scale. Again, insert box and whisker. And here we can see a much better looking scatter plot, or sorry, box plot. Okay, how many of you were able to create this box plot? Was anyone able to create this box plot? All right. So thank you, Prashant. Anyone else? Irmak? Which buttons? To create the plot, you mean? So when I create a plot, I go to insert, and then here you have this histogram, and you can create a histogram, or you can create a box and whisker. Now, as you can see, the difference between these samples is now easy to spot, right? We can see that overall, okay, so we have non-malignant, and here we have cloud and low, right? So the cloud and low is lower overall with the non-malignant, from the non-malignant. Now let's try to do a histogram. So let's take the first sample and create a histogram. Okay, so this is non-malignant. And let's now do the same thing with the first one here. And so we can see that in the histogram, move this out of the way. In the histogram, we can now visually see a lot of the data much more clearly. Okay, how do we compare them? So here we compared the interquartile range, the maximum, the minimum. We can look at the mean and the median. So these two, the dot and the line, represent the mean and the median. So we can compare some general statistics visually. But how do we know that overall samples from the same condition compared to each other? The way to do that is to create a scatter plot. So for example, we have two samples that are non-malignant. How do we create a scatter plot from both of them? Scatter plot is an X and a Y axis, right? So we can just take the non-malignant one, the non-malignant two. This will be our X axis, this will be our Y axis, and we can create a scatter plot. What does the scatter plot represent? Each dot here represents a gene. And each gene could be measured from sample number 1, which is our x-axis, or sample number 2, which is our y-axis. So here, I am expecting the bulk of our data to be comparable or correlated. So I can click on this and add a trend line. Okay, and that trend line will show me how do they compare between themselves. But the bulk of my data is down here at the bottom, so I don't really know whether the bulk of my data is correlated. Now I can do the same for this one and also create a scatter plot. And here I can see that the data is much better aligned and stretched out along this trend line. So we can change this trend line to maybe red. And maybe let's not have it. Okay, so here we have a trend line, and here we have a trend line. Okay, how many of you have been able to create these plots? All right, excellent. So this gives us a pretty good view of different statistical properties of our data and starts to show us how we can compare them between each other. So we can look at different statistical properties. For example, we can look at mean, median, maximum, minimum, and interquartile range. Or we can look at the distribution of our data. And we can also compare samples. So let's take a look. What if we compare our samples that are not from the same condition? So if you simply drag this one over to cloud and low, you can see how all of a sudden they are not comparable to each other in terms of the gene expression overall. Right? How many of you have tried this plot? I mean, I know that Irmak has, Genova, Prashant, Jadeb, Jigar, Stephanie, have you been able to visualize these? All right. Okay, so this kind of, how do we biologically interpret the dots? So the dots are genes, right? So if I take any dot here, so this is, for example, a dot, right? I can find this dot, and this dot represents a gene. So if I want to compare a single gene, let's take this gene, for example, I can create a bar plot. So here's a bar plot, and I can see that it is upregulated in the non-malignant and downregulated in the cloud and low. I can move on to the next gene, and the next gene, and the next gene, and so forth, and I can see their level of expression across different samples or across different conditions. Now, you have a good point here, but I have 1,000 genes. How do I find genes that are like this, or how do I find genes that are opposite way, that are upregulated in the cloud and low and downregulated in the non-malignant, right? How do I then interpret their biological function? So let's move on and see how to do that. So we've done some basic comparisons. Before we jump into individual genes, let's try to understand what does all of this data together mean about our samples overall. So we have here two groups, right? So we have here two groups, and overall we know that they're a little bit different, but how do we know that they're different in a comprehensive way? One of the ways to do that is to run a principal component analysis. So let's just kind of see how to do all of these steps together. We can save this data. Let's take this data, the original data that we had. I'm just going to copy this over to a new tab. And I don't need these charts anymore. So we saw that we can take two samples, or we can take all of our samples based on all of our genes, right? So for example, like this, I will see the differences between my samples based on gene data, right? So this is, sorry, let's do it like this. So based on, if I take this as X and this as Y, I can insert a scatter plot. Okay, and what do I see? I see how my samples, right? So these are my samples. Let me just clarify that. So I'll just select my series 1, and that will be non-malignant. Oh, sorry, this is going to be non-malignant. X values are going to be these. Y values are going to be these. And then I'll create a new series called cloud and low. And I'll say that the x-axis is this, and y-axis is this. So I can see that all of my samples, just based on these two genes, all of the cloud and low are close to 0, because I can see here 0, 1, 0, right? So this is very low. And all of these are actually right here, so they're very high. So based on two genes, this chart, scatter plot, represents data from two genes. I can easily distinguish all of my samples. Now, I have 1,000 genes. How do I incorporate all of the 1,000 gene information into my analysis? If these are 2 dimensions, so this is dimension 1 and dimension 2, now I'm looking at a 1,000 dimension data set, right? So this data set has 1,000 dimensions. A way to deal with such multi-dimensional data is to use what's called principal component analysis. So if I use features in my analysis, I can use 2 genes. Maybe I can create a scatter plot, and that will be 3 genes, right? So that's the most dimensions I can look at. How do I actually go to this multi-dimensional space that includes all of the genes? The way to do that is what's called principal component analysis. What it does is it looks in the data from a mathematical perspective. It can look, we cannot look, but we can just analyze that space. And we can measure the variance in this multi-dimensional space to understand low and high variance, right? So high variance will say some differences, will explain some differences between samples. Low variance means it's not explaining any information that is in this data. But finding this high variance, I can then make a projection in that multidimensional space. So that projection will maximize variance, and the vector along which I make this projection, I will call a principal component. And then I can take the second principal component, which has the second most variance in the data, and create a principal component space that explains the most variance in our data. So that will be principal component 1 and principal component 2. So let us go and run this kind of an analysis on the T-Bioinfo platform, where we will see here under utilities how to combine quantile normalization with pre-processing or log normal transformation with a principal component analysis that will allow us to visualize our data from 1,000 genes in just 2 dimensions. So let's head on over. First of all, we will save this data, right? So let's delete this, and we will say File, Save As. And I will just call it, remove the NAs from here, right? So no NAs. And instead of Excel, I can save it as a tab delimited text. So it will be a txt with tabs separating my columns. And click on save. And that's okay. It has multiple sheets, but I'm only looking to save this one. So I can click on OK. And I do want to save it as a tab delimited text, txt. Okay, does everyone have this data set saved so we can proceed? In case you don't, I'm just going to give you this text, give you this right here. Okay, so in case you weren't able to save that, you can download it. Let's go to server.t-bio.info. So to this link. Okay, let me know once you're there. All right. So there are multiple places where we can find this, but for now we will go to utilities. So if you open the utilities section, the first thing we need to do is upload this file. So you go to File, Upload. You can upload the file directly from your computer by adding files. And here I have my data set. So I'm going to upload it, make sure it's 100%, and then press on Continue. Now we can click on Start, and then Quantile Normalization. So in the quantile normalization, I can also select this to be transformed to logarithmic scale. So here I can say yes, and give it a threshold. So a threshold in logarithmic scale is going to make sure that everything under the threshold is going to be normalized. Everything above the threshold is not normalized. So if I'm expecting a high signal, that signal is going to be not affected by normalization. What quantile normalization does is it just reorganizes the data so that it is in the same order from top to bottom, but based on an alignment between distributions of our data. Okay, so here, for example, this is a logarithmic scale, so it's not going to be 10,000 anymore, so I can leave it at 5, for example. And after quantile normalization, I'm connected to principal component analysis that again gives us some options. For example, we can filter the data, which we don't need to filter, it's already just 1,000 genes. Transform into logarithmic scale, no, because we already did that. And then we have some additional options that we don't need to worry about right now. So we can just click on Save and then End. Uploading. So let me just start this real quick and then I'll do it again. Okay, so let's try this again. To try it again after you've started it, and you can see it started right here, you just click on Create Utilities Analysis Pipeline. Okay, so File, Upload. Make sure that you are doing Add Files and not anything else. So Add Files. And then here I have the table that I saved in TXT format. So make sure that you're using the TXT format. Choose. And then wait until it's 100%. Okay, yeah, so how do you convert the file type? Just from here, You do save as. Yeah, okay, great. Okay, how many of you were able to run this, start this pipeline? Genova, great. Anyone else? Well, as I explained, the threshold is based on your exploratory analysis. So if you open this back in Excel, you have to convert it to logarithmic scale. So I'll just give an example. Okay, so here if I do this real quick. Okay, so here, this, remember we can use a histogram. And so we want to keep the significant signal, which is here. And you can see here that this bulk of our data, which typically represents kind of the normal function of the cell, it's going to be, you know, maybe this hump ends somewhere right here. So this is from 427 to 549. So we chose 5 as the threshold. So right here would be the threshold. So everything above would not be normalized. Everything below would be normalized. Normalized doesn't really change it. It just aligns distribution. So normal distributions like this, if I take the first sample, and then I'm going to just take the second sample, right, etc., I will see that the humps should be comparable, right? I'm not expecting one to be dead and another one to be alive. But the significant signal, this signal right here, is going to be different. Does that make sense, Genova? Okay, so did anyone else run the pipeline? To see that your pipeline has finished, just go to My Pipelines. So here, My Pipeline Finished. And all of you can actually take a look at this pipeline if you wish. If you didn't have time to start yours, this is my pipeline. So you'll see what the outputs are. If you click on Download Pipeline Output Files, you'll see here all of our data saved in a zip file. And then also we have the results from PCA and the results from normalization. So we can just download this threshold 5 normalized PCA. And we can open this in Excel to understand what's in our table. Okay, so how many of you got the same thing? Did anyone else get the same thing? Okay. Genova, anyone else? Irmak, were you able to get your results? Okay. Make sure you don't just wait with your window open. You can reload to really see whether it finished. So let's quickly take a look. Anyone else? Prashant or J-Deb? Were you able to, or Prashant? Okay, I see Prashant has started it. Genova. Irmak, I don't see yours. So make sure that you actually have started it. If you go inside your pipeline this should be green and should say started. Okay, so press on started. All right, as you're waiting for your results, all right, looks like you did something. All right, so there we go. You started. JDeb, were you able to start this? And it looks like Jigar. Okay. Okay, so let's take a look at the results really quickly. In the results, you can see that we have principal components, 1, 2, 3, etc. And here we have our sample names. So if I take these two, this will be my X and this will be my Y value. I can just select these, insert, and here create a scatter plot. And the scatter plot will show me dots, not as genes anymore, but these are going to be our samples. So let's go ahead and make our series so that we can color them according to the group that they are in. So here I will say series 1 is going to be non-malignant. And I can take these as my X and these is the Y for the non-malignant. Create another series called cloud and low. And then take this X and this X. And here we can just add a legend. So we have more clarity. We don't need these lines. And we can say that this is PCA, PC1, and PC2. So here we have all of our non-malignant samples, and here we have all of our cloud and low samples. So the difference between them really does exist based on all 1,000 genes. How much data is explained by our x-axis? The answer is 78% almost. And how much is by second is almost 5%. So together, 78 and 5, over 80% of our variance in the data is explained by these two principal components. Do you have any questions about this? Okay, did anyone have any questions about this? Did anyone have any questions on how to run this? All right, so for the sake of time, we don't have that much time left. So let's turn to statistical analysis. This was exploratory analysis. I now understand what the majority of my data tells me. But how do I find the specific genes that can illustrate this difference between these two different sample types? Let's go back to our original data set. So this is our original data set. And let us create a statistical test. Okay, the statistical test will be for our observations. I'm just going to extend this a little bit. And someone in the beginning here mentioned that I can do a t-test, right? So what does a t-test do? The t-test is a comparison between means of groups, also incorporating the variance within each group. So it is the mean divided by the variance. What that tells us is that if I have a sample that is not very clear cut, okay, for example, let's put here Okay, let's say a sample like this, where I have overall the genes are upregulated, but here I have some examples that are also upregulated. I can ask the question, well, what does it tell me about the statistical significance or the confidence that I have in determining the difference between them? So if I take the variance within this group and the variance within this group, and I will incorporate that into my statistical analysis, I know that I will find only the differences between groups that are statistically significant. So to run a t-test in Excel is also very easy. I can do t.test. And then array means just a collection of numbers. So I can just take this array from non-malignant. That will be my array number 1. And then from cloud and low, that will be array number 2. And here we have different types of distributions. So we have a two-tailed distribution. And here we have equal or unequal variance. And we don't know what, I don't think is equal. So we're going to have unequal variance. The output of t-test is a t-statistic. But the t-statistic, which is a distribution of probabilities, could be used to infer the p-value. And the p-value is just the area under curve of the t-distribution. So this actually in Excel, the p-value. And the p-value could now be sampled for all of our observations. These are our observations. And we will see that the value could be used to find here only those, sorry, let's do it like this. And we can find here only those that are statistically significant. Okay, so you can see that all of these values are actually statistically significant, except for this last one, right? So let's see, why is the last one not statistically significant? Okay, so here you can see an example of a bad gene, a gene that is not differentially expressed. And the difference between them, right? So the first 5 are normal-like, and the last are cloud and low. And you can see that because I have these 2 zeros right here, it's not very clear what is in this group. And so therefore, the difference between conditions is not statistically significant. Okay, does that make sense? Does anyone have any questions about t-test and statistical analysis of this data set? What do we conclude from the p-value? So p-value helps us test the hypothesis. The hypothesis that we're testing here is whether the gene is differentially expressed between the 2 conditions. If it is different between the 2 conditions, we have a good p-value or a low p-value below 0.05. If we have if we disprove our hypothesis, that means our p-value is going to be bad, or the p-value is going to be above 0.05. So here, above 0.05, 0.9, right, this shows us some questionable difference. But a good p-value, like this one, gives us fairly clear difference between this group and this group, right? A really good p-value, we go to the very top, so a really good p-value shows us a very clear difference. Upregulated, downregulated, non-malignant, cloud and low. Does that make sense, J-Deb? okay, how do we actually do this kind of an analysis in a proper way? What do I mean by a proper way? Well, there are two major issues that we did not address here in this simple Excel analysis. The first one is we used just normalization using logarithmic values, and that doesn't mean that we accounted for some artificial variation that is only preserved in the raw data. So we need some more advanced normalization before we run this kind of an analysis. It's actually quite obvious because all of our 1,000 genes are differentially expressed. The second problem is the problem of what's called multiple testing. When we do multiple testing, we have a high probability of finding false negatives and false positives just because of the number of observations that we have. So if you imagine, if you want to test all the people in China and have them flip a coin, you are very likely from the 3 billion people that will flip a coin, you will find some people that will have all of the coins flipped just to one side. What does that tell you about the coin being faulty or good? Really nothing. It's just because so many people did the test, some people will likely be on the end of that distribution. And so we really need to adjust for this multiple testing artifact. To do that, we have what's called a differential gene expression analysis. Okay, so let's go back to our areas of analysis. And here's a data set that you can use for this analysis, so it actually comes out good. So let me get, sorry. Okay, so here I'm just going to sorry. Let me just find this data set. I forgot where to put it. Oh, okay, I know where it is. Okay, there we go. So we're going to take this data set right here. Okay, so please download this really quickly so that we can run this pipeline together, and we'll look at both statistical analysis of differentially expressed genes and functional annotation of these genes. Okay, so here we have two types of samples, ER positive and triple negative breast cancer. Please let me know once you have downloaded this file. All right, so let's go here into this differential expression section. And you can click on File Upload. I'm going to select this file. wait until it loads to 100%. Okay, let me just give it a pause here. So those of you that want to follow along, just let me know that you got there and you have the file ready. All right, thank you very much. Where's differential expression? So differential expression is right here under data mining, differential expression. Okay, again, file upload. So upload data. Choose that data set. Okay, JDEV, did you find it? All right, excellent. Okay, and now we'll click on group selection. So we need to compare two different conditions. So we can drag all of the ER in group 1 and all of the TN in group 2. So we have 4 samples in here and 4 samples in here. Submit. And now let's create the DESeq2 pipeline right here, which will give us a difference between the gene expression and annotation of those genes. So here you can see some options for filtering, the threshold, and the database. And so let's keep all of this the same. So just click on save right here and run the pipeline. Right here, run the pipeline. So it should say started when you run it. Okay, let me know when you have started this. You can see that once I started it, it comes up here on the activity tab. Okay, let me know once you run it. So Genova started it. So if you go to my pipelines and you've started, it should say processing. All right, Prashant also. JDEV, were you able to figure it out? All right, great. JDEV, Prashant, I don't see yours yet, but no files. Well, did you upload the files? I see. Yeah, so you put in the right files. All right. So let's take a look. Okay, what is going to be the output from this analysis? So I'll just reload the page. Okay, reload it again. All right, so 100% done. Let's see what is the result. So in the results, I have a few different outputs. The main output is this one. So it is the significant genes by p-value. So we can just download this table. So open it in Excel. what do we have here as a result? As a result, we have the full table of expression, which is right here, right? So this is the full table of expression. But we also have the ensemble ID, entrée ID, the symbol, the common gene name, and the full name, as well as some statistical properties like the base mean, which is the mean for the samples overall. And we have the p-value, the p-adjusted value, which is the adjusted p-value for false discovery rate, and the log2 fold change. In other words, if I find a gene, let's see a good p-value. So this first one is a good p-value. Let's take a look at what it looks like. So this gene is expressed, mesenchyme home box 1, is not expressed in ER positive, barely expressed at all, right? This is at 0. Expressed in triple negative, but in 2 it expressed high, and in 2 it's expressed not so high. Overall, it has a good statistical p-value, just because we only have 4 samples each, so yes, it's likely to be upregulated in the triple negative. And we have a fold change. What is the fold change? The fold change is if I take the average between these and divide it by or divide these by these, or these by these, doesn't matter which group by which, I will have a number by which I have to multiply the average to get to here, right? So that's the fold. How many fold is this one expressed higher than this one? It's this number divided by this. Okay, so here I can sort through. Let's create here a filter. I can make sure that all of my adjusted p-values are going to be less than 0.05. And all of my log fold change is going to be, let's first take a look at greater than 2. So all of these genes, you can see that they're all upregulated in the triple negative. How many of them? Not so many, right? But I have here a number of genes that are upregulated only in triple negative. You can see the higher numbers are all on this side. If I want to say that they are less than -1, I have 2 genes that are upregulated in the ER positive and downregulated in the triple negative, right? So I can calculate how they are different. The other thing that you will find here are some visual summaries. For example, you can take the volcano plot and you'll see what are some visually differentially expressed genes. So this is a PDF where you can see the differentially expressed genes. The majority of them, right, are right here on the triple negative side. So they are upregulated on the triple negative side, downregulated on the ER positive side. You can also find in the outputs the significant pathways. So pathway analysis, right? So you can see how many of these differentially expressed genes are present in which pathways. Okay, so unfortunately we ran out of time, but I hope that this gave you a little bit of an insight into how to do exploratory data analysis, so understanding statistical properties, comparing different samples and different genes, and then understanding the biological interpretation. What does this actually mean when we find differentially expressed genes, and what is their biological significance? So to speak about the specifics of how do you apply this in the context of a project, we will talk about it next time to discuss different project examples that you can use to really understand how this all works together. Now, before we leave, are there any quick questions? If you have some major questions and concerns, you can post them on the forum. We'll be sure to address those.
If not, we will continue speaking about projects in our next session.